So today's lecture is going to be a bit different than the previous lectures, as we are not going to discuss a imaging modality per se. However, I feel that the topic of understanding how traces or contrast agents, how they enter the tissue and how we can understand their appearance in the tissue is of wide-ranging importance. It's also important for processes well beyond biomedical imaging, such as pharmacokinetics, etc. And therefore, I wanted to spend this small segment this week on understanding the kinetic models and how we obtain metabolic information or information on the processes. And so here the first question is, why is it important to understand basic modeling of linear systems? So let's take a situation. It's here presented as a hypothetical situation, but it actually does have some relevance to what I've experienced in my career and what is not infrequent to happen. So we'll take a situation here where we have a team A, a research team A, that measures a high expression of the message of a gene, the mRNA. Team B measures at the same time point a low protein level. A year later, Team C reports a low mRNA of the same system, whereas Team D finds a high protein level. So the question is this a controversy or is there a common underlying explanation? Trust me, looking at these things as they reported for research teams, conflicting findings, this does give rise to quite some controversy at scientific meetings. So let's try to understand this situation. I'll plot here on the x-axis. We'll have the time here. And now what we're plotting here in red is the expression of the gene. That's the mRNA as it has been measured. Or let's say this is what's happening in the system. And then let's look at the protein level of that particular gene, so the protein that's being expressed. Those two time courses are not the same. So what we have here now is situation 1 is where the measurement was done at this time point. Situation 2 corresponds to a measurement at a later time point. So if in the discussion of these seemingly controversial results we omit the factor time here on the horizontal axis, we can very well enter into considerable controversies that are actually, if we look at now this graph, we're not looking at a controversy, we're just looking at different time points. The system we're discussing here is DNA to expression, the mRNA, and finally the translation to the protein. And what we're looking at here is essentially a biochemical reaction. We have the precursor, that's the mRNA, and we have the product, which is the protein. Clearly, we cannot have protein being expressed before we have the gene being expressed. The protein is not synthesized before we have the signal in the cell that this protein should be expressed. So there's a causality issue here, and the mRNA has to precede the expression of the protein. So this is situation 1, and this is situation 2. Two different time points. If we look at it from this standpoint, then actually our conclusion must be there is no controversy, we've just been looking at different time points. Now, the system of product-precursor relationship can be described mathematically. We can set up a model, and the underlying mathematical principles, as we'll see in this lecture, they're actually fairly general applicable. We can apply them to contrast agent dynamics, uptake of contrast agent. So instead of mRNA, we have contrast agent in the blood, and instead of protein, we have contrast agent in the tissue. Enzyme kinetics, we have the reaction velocity that goes up first, for example, and the product buildup will come later. This cannot happen in the other way around. Take diabetes as an example. We have insulin. Patients with diabetes very often have to inject insulin, and the effect on glucose uptake is delayed. So insulin first, then changing glucose uptake predominantly in the muscles comes later. Sailing. For those of you who have experience with sailing, you know very well that if you change the rudder or the sail position, the change in direction or the velocity of the boat, that does take some time. Economics is also an example. If we change the financial incentives in the system, until that changes the production and the availability of the product in the market, that is also a relationship, a causal relationship, where we have a delayed response to the input that's changing. So the underlying principle here, in all cases, it is about inertia. It's resistance to change. Things don't happen instantaneously, and this is the concept of inertia. We've had, for those of you who remember your freshman physics course, we have talked about inertial mass. In those courses, mass is a resistance to change the velocity, Newton's second law. Also there, the concept exists in there, and this is taken here in the example of sailing. Of the techniques that are highly dependent on these description and on a good understanding of the kinetics that are happening is the FDG-PET technique. And this is a technique to measure intracellular glucose metabolism. So before I go into the specifics of kinetic modeling, I want to just give you an overview of the processes involved in measuring intracellular glucose metabolism using fluorodeoxyglucose or deoxyglucose. Now, the tracer molecule here, FDG, is taken up into the cells, and this uptake depends on the expression of a protein called GLUT. GLUT here stands for glucose transporter. So, glucose cannot go across the cell membranes. It needs a protein, sort of like a channel, that allows it to cross the cell membrane, and these are the glucose transporter proteins. It also depends obviously on the glucose metabolism. The uptake depends also on the activity of the first metabolic step of glucose metabolism, that's glucose phosphorylation, mediated by the enzyme hexokinase. So we'll look here at the cell. This is the cell membrane. We have here our glucose transporter molecule GLUT1. This is one isoform of many isoforms. There's roughly 9 glucose transporter isoforms that have been described in the literature. GLUT1 is the one that's present in the membranes of red blood cells and in most cells in our body. Here's the molecule, that's the fluorodeoxyglucose. We administer it, it comes to the cell membrane and then traverses the transporter, and thus the FDG molecule ends up inside the cell. This transport into the cell can happen by GLUT1. It can also happen by other isoforms, such as GLUT3, GLUT5. GLUT3 being the isoform at the neurons, and GLUT2, for example, being the predominant transporter in the pancreas, important for diabetes. So now we are in a situation that we have fluorodeoxyglucose inside the cell, and now comes the first metabolic reaction mediated by hexokinase, and this process happens here in attaching a phosphate at the 6th position, thereby producing fluorodeoxyglucose 6-phosphate. This molecule now has interesting properties. Number one, it cannot be further metabolized by the next enzymatic step in glycolysis, so glucose 6-phosphate isomerase does not metabolize deoxyglucose 6-phosphate, so this does not go on. And glucose 6-phosphate or deoxyglucose 6-phosphate is not transported out of the cell. So, in a first approximation and a simplification of the reality, we have fluorodeoxyglucose 6-phosphate or deoxyglucose 6-phosphate is now metabolically trapped inside the cell, and this is what how FDG-PET or deoxyglucose methods measure glucose metabolism. Now, to give you an example here, we'll take actually not FDG, but we'll take another deoxyglucose, the C14 or tritiated deoxyglucose, And the technique used here, the examples I'm giving, is autoradiography. So what we're looking at here are images of the brain in grayscale. These are brain slices, so the brain has been harvested, cut into thin slices. We see on the left, we see the images, and on the right in the color code, we see the glucose metabolic rate, with warm colors being a higher glucose metabolic rate and cool colors being a lower glucose metabolic rate. So these brain slices are placed on a radiographic film, and then the emission of the radioactivity from C14 and or tritium, those are beta particles, is being recorded on the film, and this gives us the color-coded images. This is autoradiography of a rodent brain, and here is an example, a close-up. This is showing the columnar organization of the whisker barrel cortex, where each of those columns corresponds to a specific whisker with a higher uptake. So this is a very powerful technique to measure glucose metabolism, and we will use it as an example throughout this lecture 7, and in the end explain how the FDG technique functions. So first, however, I want to introduce some basic principles. First of all, the question, what is a compartmental model, a compartment model? And this asks the question of what is a compartment. So we'll take the definition of a compartment here, that we'll take the concept that we have a physiological system, and we'll decompose it, model it into a certain number, n here, given interacting subsystems. Those subsystems are typically a given chemical species in a physical space. That physical space is typically called compartment. So we have, when we give deoxyglucose, we have glucose in the blood, that's one compartment, glucose in the cell, that's another compartment, and then we have phosphorylated glucose, that's the third compartment. We'll make the assumption here that the tracer is considered to be uniformly distributed in a given compartment. There are very elaborate experiments that would allow us to say something about where in the cell this particular compound is located, but they're very complicated experiments, and for practical purposes in the context of biomedical imaging, this is a assumption that one makes. So what are the key elements of compartmental modeling? Let's look at some major elements. First, the first one is, the key element is, we're going to try to make a prediction of what's happening in a part of the body that we have no direct access to. So these are inaccessible features of the system. With deoxyglucose, this would be intracellular glucose metabolism. So the inaccessible portion is something inside the body, inside the cells, and we want to get some ideas of what's going on here. So for example, we could say we have a model taken from biochemistry or metabolism. We have a chemical species A that interconverts into a species B and is also being converted to species C. That would be an example here. The second element of compartmental model is that we do the measurement in an accessible portion. So what is the accessible portion? That is essentially the measured output. And what do we have access to? Well, we typically have access to the blood vessels, so we can take blood samples. And we typically, in imaging, we have access to the measurements that are being done with the imaging technique, that is, to the value of the pixels. So those are our accessible features of the system. So we'd like to have, from the measurement in the blood, from the pixels, this is our output, we'd like to say something about what's going on here inside our cells. We have also, of course, a known input into the system. So this is a given example here of what's happening in the blood. We inject here something very rapidly, and this compound is decreased. This can be a tracer, this can be a contrast agent. So this part here, that's our accessible portion. The blood measurement is here. And then, based on the measurements in the accessible portion, we're going to try to estimate specific parameters of interest. One example already mentioned is the metabolic rate of glucose. So here is an example we have fluorodeoxyglucose in the blood, in the plasma. We have the transport into the cell. Then we have the free deoxyglucose or free fluorodeoxyglucose inside the cell. And we have then through the glucose metabolic rates, we have the fluorodeoxyglucose 6-phosphate or deoxyglucose 6-phosphate. So these are the three compartments: blood, brain, the species. Metabolically, we have three compartments: free glucose in the blood, free glucose in the tissue, and phosphorylated glucose in the tissue. Now, modeling, assessing the kinetics, we're making steady state assumptions. And there are two assumptions here that are important to mention. One is that the metabolic rate of the process is not changing with time. So we are assuming that, for example, the rate of glucose metabolism inside our cells as we do the measurement is not changing. It may change, but what does it mean? If we make this assumption, it means we're calculating an average value of glucose metabolic rate. We're also assuming, as a steady state assumption, that the concentrations of all the compounds involved and the enzymes, etc., are constant during the evaluation period. With these two steady state assumptions, we can actually have a relatively straightforward mathematical description of what is going on, and that is, as we shall see, that we can describe the processes, the metabolic processes, with first order rate constants. They're now not true rate constants, so we will call them pseudo rate constants. Before we go into the specifics of the mathematical description of predicting what's going on inside the cell based on what we measure in our pixels and based on what we've measured in the blood, I want to first introduce, however, a technique that was used more than a half century ago that also allows a rate determination. And here the principle that is being used is conservation of mass. That already allows us to make some statements regarding what's going on in the organ of interest. And this is based on the Fick principle. So let's consider here the situation. We'll draw here the tissue. That's our organ of interest, or the tissue. We have, of course, blood vessels that supply the tissue with nutrients and oxygen, and that remove the metabolic end products. So this is a perfused organ. This is the blood, and here we have the blood that flows, and its flow is indicated by the parameter F. So as the blood flows, it flows through the tissue. The number of molecules that goes in is equal to the number of molecules that goes out, if this molecule is not being used by the tissue. So now let's assume that we are looking at a particular molecule here. We'll just stay abstract for the moment. Here, these are the molecules that are coming in, and now the tissue is taking up these molecules, taking it up, storing it, or taking it up and converting it to something else that we don't measure. So an example is, if we take oxygen, oxygen 15 we've seen last week is a PET tracer. So if we take oxygen 15, molecular oxygen 15, we breathe it, then we get the oxygen here coming in, and what goes out is H2O15. We can say the same thing with other compounds. So now we'll define this consumption of this particular compound, X here, I'll call it X. We call that the metabolic rate of X, or MRX. That's the metabolic rate in the tissue, the rate at which this compound is being consumed. Now we'll have a certain concentration of this compound on the supply side, so on the arterial side, that is on the blood entering the tissue. I'll call this concentration X in, and we'll have a concentration of this compound in the blood that leaves the tissue, and we'll call that X out. And so now what we have is the blood supplies a certain number of molecules per second, a certain number of molecules per second or per minute is being removed, and so fewer molecules per minute are leaving the tissue, are leaving by the bloodstream. And if we now look at mass conservation, this simply dictates that the number of molecules, that is the metabolic rate, that are being consumed is equal to the number of molecules coming in, that's blood flow times the concentration of blood entering the tissue, minus the number of molecules leaving the tissue, that's blood flow times the concentration of blood leaving the tissue. So this gives us the metabolic rate of this compound. Now X here, to give you something specific, X here can be oxygen. So we have oxygen coming in, oxygen leaving, and oxygen being consumed. It can be glucose, it can be ammonia, or it can also be water for this principle. Now let's look at brain physiology as an example. We know from physiological experiments and by experience that if we are in a dark room and we suddenly look, for example, at an image, we have in certain regions of the brain a quite strong increase in blood flow, but hardly a change in oxygen consumption. So oxygen consumption does not increase in parallel with the blood flow. So what is the consequence? Think about it. So we're increasing the blood flow much more than the consumption here, the metabolic rate. You can think of it, take the extreme case. Let's assume that the blood flow is increasing, but the metabolic rate is not changing. So we have no change in oxygen consumption, but we have an increase in blood flow. If we have no change in metabolic rate, but this increases, the blood flow increases, then the difference in concentration between what's happening in the supply side and the output side in the blood flow, this difference has to reduce. So now, the arterial concentration of oxygen is not going to change. There is no mechanism by which an increased supply will change the concentration of oxygen of the blood entering the tissue. So by consequence, the only thing that can happen here is that the venous effluent of the oxygen concentration in the venous effluent from the tissue is increasing, and so therefore the concentration difference between in and out decreases. So we'll take this particular equation here, we'll rewrite it in terms of oxygen. So oxygen concentration entering minus oxygen concentration leaving is equal to the rate of oxygen consumption divided by the flow. This hardly changes, this increases, this must be reduced, this doesn't change, so the oxygen concentration in the veins has to increase. And this, as we'll see in a few weeks, is a major biophysical basis of a major imaging technique to study brain function, namely functional MRI. So here I want to bring in some definitions. We'll talk about the tracer. Classically, a tracer is a radioactive molecule, so it's emitting radioactivity. It is labeled with the isotope. It is typically linked to the molecule that we wish to characterize, or sometimes even identical. So the natural substance is called tracee here, and that would be one example we've already seen. That's glucose as the tracee, and the tracer would be fluorodeoxyglucose. But we can also have the native molecule, so that the tracer is equals to the tracee, and that would be oxygen, the example of oxygen 15 PET. Now the term tracer also implies that the substance is administered in trace amounts, so that is very small compared to the concentration of the tracee. So in the case of deoxyglucose with PET, we can introduce very small amounts of fluorodeoxyglucose on the order of nanomoles or micromolar, whereas the glucose concentration is in the range of millimolar. And this has the advantage that the introduction of the image contrast generating molecule does not perturb the process that we wish to measure. Now, we have the situation that the tracer molecules, they're not all going to contain the isotope. Some contain also the cold isotope, so they're not radioactive. And so what is important here is to look at the specific activity, and that is simply the ratio of hot to cold tracer molecules. since the cold tracer molecules are predominantly dominating the molecules, this gives us a percentage of the molecules that are emitting radioactivity. This specific activity is always measured. It's typically measured in megabecquerel per micromole or millicurie per micromole. So it's given in a per concentration unit, and we have to know it. So the measured radioactivity is converted. The concentrations in the tissue and in the blood are converted to mass units so that we have the correct physical units. In all the discussions that we have today, we are going to neglect physical decay. That is, we're going to assume that the natural half-life of F18, which is 110 minutes, that we've taken this into account, we've corrected for the fact that without anything happening, the signal intensity, the specific activity will decrease with a half-life of 110 minutes.